I'm Jiannan Liu from Indiana University, doing my PhD here. So the topic for today's presentation is the text mining and portal development for gene-specific publications on Alzheimer's disease. This is a recent work we have done recently. Yeah, here is the outline of my presentation today. Just briefly go through the backgrounds, study design, data collection steps, the data cleaning and processing, and then the portal development. So a little bit of background for the Alzheimer's disease. Alzheimer's disease, or we call it AD, is one of the most common neurodegenerative disease. So as you can see here, so it's like it's much more cases each year for 100,000 patients in the US, comparing to the Parkinson's disease and also other common neurodegenerative diseases. So the pathologies for the AD is like still not very clear, but there are two hallmark pathologies for the AD development. One is a beta amyloid plaque deposition, and the other one is like the tau deposition. So those are just some proteins that deposit in the neurons and cause some bad effects on the brain functions. So the AD is mainly impacted the senior group of people. So you can see for all the AD patients, the people who are in their older age is like largely impacted. The cure or even the slowdown of the progression of the AD still remains a very challenging problem. And so for on the publication side, this number is coming from the PubMed database. It's like there are 14,000 publications related to the AD is published in 2022. So there is a huge amount of publications in this area, especially in recent years. There are more and more publications coming out, and the number of natural language processing research has been focused on the AD, but they are mostly focused on the ontology part, like the entity identification and those graph analysis. And one project we did before is called CGP Cancer Gene and Pathway Explorer. So in that project, we also included some natural language processing pipelines to identify the information for the cancer research, trying to categorize the publications and provide some insights on the publication trend in the recent years. And the research survey and drug target identification is challenging for the AD research right now, because we even don't have a certain or very well-performed target for the AD treatments. So that's why we come up with two objectives of the study. The first one is design and implement a natural language processing pipeline to extract the gene-specific and also AD-related publication information from the PubMed database. And after we process the data, the second objective is to build a web portal to allow users to query the extracted gene-specific information easily. So these two objectives will help with the target identification for the treatment of the AD. So with two objectives, we designed a study workflow, something like this. We have different steps. First, we build a gene list. Those genes in the list are all from the RNA-seq expression data sets. Because for the sequencing technology, usually there are 20,000 different genes can be identified. So we use those common, like 20,000 different genes that can commonly identify by this RNA-seq as our initial pool of the genes that we try to identify in the publications. And then we, according to the gene list, we collect the publications for each gene. And then we build something called gene-specific publication profile. Basically just a set of the publications which their title or their abstract mention the current gene. A problem we address here is a gene alias because people refer to genes by different names, and so gene name is kind of an ever-changing property of the gene. And then we did some filtering, like gene-based filtering. We just want to make sure that the gene is occurred in the title or the abstract. Just try to give some accurate information for the information extraction. And then we have to make sure that the disease we are focusing on, like AD, is occurring in the title or abstract to make our publication profile more accurate. Then after some basic filterings, we extracted some important information for the AD researchers. Some examples like differential type, keywords, like there are some hypotheses in this field, and also there are some mechanisms, like the name of those mechanisms. Those words are collected from experts in this area. And also the brain region, because AD is will impact multiple brain regions. So some people may focus on some certain brain regions. And also one special thing we have is the mouse models, because people do experiments. It's interesting whether some certain genes is related to, whether there are some like certain mouse models is like impacting some functions of genes, certain genes, I mean. And also the co-occur genes, like gene co-occurrence in a certain publication. After we process data, we have like a final huge data matrix, and then we incorporate that into the database. Then we build a UI, user interface, for the data we collected, and did some realizations based on the data we have. The first step we have is like data collection after we get the whole gene list. The profile we build is called the gene specific publication profile. So basically what I'm talking about GSPP is like a set of CSV or TSV files, which includes a list of the publications. For each publication, we only collect the title, abstract, and some other basic information like the PMIDs. And we only analyze the title and abstract in our study. So during the process, we use multiprocessing because there are millions of publications in the PubMed, and we query each gene separately and then build this profile. This is our initial data we have, and there are almost like 19,000 GSS is built after the first step of the data collection. After we have original data, we did some filtering, as I mentioned earlier. So the gene-based filtering is just trying to make sure that the gene name is occurred in the title or abstract. The rationale behind here is like, if this gene is mentioned in the title as abstract, so it's highly likely that this gene is talked about in the main body of the paper. But if we have this gene in the main body of the paper, but it's not likely, or it's not very likely that this paper is about this gene's function or this gene's like a mechanism or something like that. And then after the gene filters, this step we address the gene alias, as I mentioned. So it's an ever-changing property of the genes. So there are some like very important genes have different names, but different papers refer them with different names. So we try to address it. And we also try to address this problem here. And excuse me. So another filtering we did is a disease-based filtering, because the mechanisms for the AD processing is kind of similar to some similar neurodegenerative disease. So we include 8 different diseases here. Here is a list of all the diseases we included. So we try to extract the information from the title and abstract, trying to make sure that these diseases are occurring in the title or abstract. And after these 2 steps of filtering, we have the final, not the final, but some like a much cleaner GSPPs in our data set. And with the data set, the next step we did is like data processing. We just try to extract some useful information for the AD researchers to look at. We have some categories here. The year is it's like a very straightforward from our initial data, because we have the publication year for each publication. And then the dementia type, we have 8 different dementia types in our data extraction step. And we have the brain region information. 13 different brain regions are in our extraction. Like and then the mouse model, we identified currently, like there are 11 different mouse models for the AD disease. So we try to identify this information in the publications. And the keywords, with the help of experts, we identified 31 different keywords in this research field. which are very popular in the research process right now. So we try to extract those information. Here is a count of all the publications includes a certain category information here. Yeah. So with all the information we have, sorry, excuse me. So this matrix you can see here, this is just part of the matrix we have in the final step. Those are like a summaries of the information we extracted. So the DEM type here is like a dementia type. We have 8 different dementia types. For each gene, we will have a role here. Each role is a gene, and we will just count how many publications mentioned the AD, mentioned the frontotemporal dementia or Lewy body dementia. And then we have another column to record the PMIDs for a certain disease here. And for other categories of information, we do have the similar structure. Basically, what we did is just trying to accurately categorizing the publications into some subcategories. And then we provide this kind of information to the researchers. So this will help them to kind of do a research survey, because when people propose their targets for Alzheimer's disease, they try to do a research survey, and then they try to find all those papers related to the gene, and also some certain topics they are doing their research on, and then this kind of information will help them to identify those publications. Then after we have the final huge matrix, we develop our web portal for the data we have. The tool we call PubAD, publication AD basically. The tool we use for building the web portal, something like Bootstrap to control the layout of the page. The visualizations, the charts we have in our portal is developed by D3.js, and the backend database we use is PostgreSQL. So here is a query for the APOE. APOE is like a very hot gene in this research field. So if you query this one, you can see the publication trend and some other basic information about the gene. And we made several like panels in the results. For the first one, as shown here, it's the keywords. Like all those keywords are like identified by experts, and then we plot a word cloud plot with the publication, like a number of publications. The word cloud is based on number of publications for each keyword. So people can clearly see which keywords is mentioned together with the current query gene. And we have some like dementia information and brain regions and co-occurred genes, like gene co-occurrence. As you can see, the OSM is occurred. Okay, this query is from the STAT3 gene. So the STAT3 gene is occurred the most with the OSM gene. Those kind of visualizations can be provided by our web portal, and also all the bars here on the like bar charts are clickable. So you can click the bar, and then you go to the PubMed website to check all the publications recorded for a certain category. And that should be all we have done for our study. So yeah, I'm ready for any questions.